have the cell cycle. The cell cycle is composed of interphase, which is where most functioning cells spend most of their time, and especially in the G1 phase. And that is where cells are usually held when they're just carrying out their specific function designed to them. When cells receive a signal telling them to divide or that something has gone wrong, they will then go on to the rest of interphase, which is S phase. But before they go on to the S phase, they need to pass the first checkpoint, which is the G1/S checkpoint. In the S phase, they synthesize proteins that will then later be used throughout the rest of the cycle. After the S phase, the cell goes on to the G2 phase, and here there is last-minute preparations for mitosis. The cell will not continue on to mitosis until mitosis-promoting factor signals the continuation, and they get through the G2M checkpoint. In the M phase, or mitosis, they go through a series of steps, including prophase, prometaphase, metaphase, anaphase, and telophase. In mitosis, we begin with prophase. During prophase, there is nuclear material condensing and forming chromosomes. The chromosomes, sister chromatids, which compose chromosomes, are held together by cohesin. Then after prophase, the cell goes on to prometaphase, which is where we have fully formed and condensed sister chromatids, and they are held together at the spindles by kinetochores. After prometaphase, we then go on to metaphase. During metaphase, all the chromosomes have been lined up in the middle, and they are held there by the kinetochores. They will not go on to the next phase, which is anaphase, until they get through the checkpoint and anaphase promoting factor signals that it is okay, and after all the chromosomes have been lined up in the middle. Afterwards, they continue on to anaphase, which is where sister chromatids are separated to opposite sides of the cell, and they then go on to telophase. In telophase, there's two daughter cells identical to the mother cell produced, and there's a cleavage furrow formed as well. In plants, instead of having a cleavage furrow, they produce a cell wall. Another step or cycle is meiosis. Meiosis is similar to mitosis, but there are several different factors. In this particular one, this is depicting meiosis 1. Meiosis is composed of meiosis 1 and meiosis 2. During prophase, as in mitosis, we have DNA condensing, forming chromatids, but here we actually have the chromosomes from the mother cell and the father cell condensing on top of each other, actually forming tetrads. The cell then goes on to metaphase 1, which, as in mitosis, is the lining up of the chromosomes in the middle. After metaphase, the cell goes on to anaphase 1, and the difference here, as in from mitosis, is that the sister chromatids are actually held together, but what is separated to the opposite sides of the pole are the chromosomes, the homologous chromosome. After anaphase 1, the cell then goes to telophase 1, which has the forming of the cleavage furrow, and we actually have two different daughter cells produced. Unlike mitosis, where we had identical clone cells to the mother, here we have half mother cells, half father cells, and that is due to the crossover of DNA happening in prophase 1. So we have DNA crossing over each other, and that produces two different cells. Now, meiosis 2 is the exact same thing as mitosis. The cell will go through the same process, but we will end up with 4 different daughter cells. Now, some other differences between meiosis and mitosis is that meiosis is mainly for producing gametes or sex cells, while mitosis is a cycle of cells to produce more cells, growth, and reproduction. As mentioned before, mitosis produces two daughter cells cloned to each other and cloned to the mother, while meiosis produces four cells, each with a different genetic markup, half coming from the mother cell and half from the father. Another difference is that meiosis produces haploids while mitosis produces diploids.